Hi, my name's Miss Samantha Conroy. I'm a clinical research fellow from Sheffield, and I've been asked to do a short video just to explain a little bit about my research that I've been completing over the last 12 months. And that's been a part of my Tough Research Scholarship Award that I was awarded in 2020. Now, the title of the project is Ex-vivo high throughput drug screening in bladder cancer, which sounds really complicated. But what it boils down to is identifying patients who've got a known bladder tumour who are going to have that tumour removed surgically. And this is a normal step in their care. And what I ask is that when we take the tumour away, would they be happy for me to take a small sample of their tissue up to the laboratory for additional testing? And the prime means of doing that is that we break down the tumour tissue into all its individual cell components. So this might be normal cells, immune cells, and actually the tumour cells. And what we do then is we seed them onto a very large drug plate, which contains standard of care drugs that we receive in the clinic, some new and novel drugs that might be in clinical trials, and then some other drugs that are used for different types of cancers. And that's to see which ones work best for each individual patient. The reason that we need to do this is twofold. So first is that we know that if we have all bladder cancer patients put together in one category, they receive a treatment, and actually we know that all of them don't respond the same. And that's because inherently the genetic makeup is different, but it's hard for us to identify that in an NHS cancer care pathway. The second is that we want to identify new treatments for patients with bladder cancer, because despite us knowing a lot more about cancer and how we treat it and how we detect it, outcomes for patients with bladder cancer, particularly advanced bladder cancers, has not improved in the last 30 years. So what we wanted to do was see whether we could use this model to identify responders and non-responders to standard treatments, and also whether we could identify any new treatments for patients with bladder cancer. So across the course of the last 11 months, despite the COVID restrictions, all of the patients were incredibly positive that I approached about the study. And this meant that I recruited 82 patients to the study. And I must thank them all for their involvement and participation through such a challenging time. Of the 82, 52 patients had enough tumour tissue for us to run the ex-vivo testing. And of those, we generated 44 endpoint results, which means that we generated a drug response profile for 44 patients. And one of the exciting things about this study is that we actually identified a standard drug that we know all the toxicities for, that actually was effective for every single patient. So something that could be added immediately to their care if they were to need additional treatment. And the second thing was, we actually, for almost all patients, identified a new drug that would be effective against their tumour cells. So again, one of the successes of this, the initial stages of this study, is that we've developed a method which may help us develop drugs quicker and hopefully get them into the clinic to start treating patients. I must say though, this is the early stages of this research, and there's a lot more that needs to be done. So there are certain parts of our kind of laboratory protocol that need to be worked on and optimized. And that's something that I'm looking to apply for further funding for. And then in the future, what we really want to do is generate enough data to run this in a clinical trial. So can our model better predict patient outcomes and direct treatment than standard cancer treatment pathways that we have in the NHS at the moment? So that's the longer term goal. I'd just finally like to say thank you to all the participants once again. Thank you to my supervisors and my support that I've received from Sheffield Teaching Hospital and the University of Sheffield. And most importantly, thank you for all the support from Tough. It's been an absolute pleasure working with such a great charitable organisation, and hopefully I'll do more research with them in the future. So thank you very much, and I hope to see you all soon.